Okay, so another important aspect of the late stage um, secretory pathway is that seen here from these images of pro-insulin antibody and insulin antibody experiments. So once insulin is produced in the Golgi, or in the once it is modified in the Golgi, it is released from the trans Golgi network in these vesicles, these secretory vesicles. And notice that they're labeled here with antibodies. The this proinsulin, which is a immature form of insulin. They aggregate into these vesicles in large number, and we can see from this other image here that once the uh, proinsulin is converted to insulin, we could use insulin antibodies to detect that the insulin has been created from proinsulin, and so this basically proves that in, uh, proinsulin is released from the trans Golgi network, and they are modified inside their secretory vesicles to produce insulin. And so usually insulin is released upon some sort of stimulus, like some chemical signal. Usually when there's a high level of sugar in the blood, of glucose, these insulin secretory vesicles will be released through the plasma membrane into the bloodstream by beta um, by these specialized uh, beta pancreatic cells. And because there's a lot of proteins that are made or that are transported through the Golgi, um, especially like lysosomal enzymes, uh, they have to be they have to be inactivated during this transport so that they do not interfere with the function of other proteins in during the transport meant for other parts of the cell. And so they have to go through a lot of modification in the through the through this proteolytic processing of proteins uh, in constitutive and regulated secretory pathway. So you can see here in the constitutive secreted proteins, this proalbumin, um, it is operated on by furin endoprotease, which cuts the amino acid polypeptide into two different strands, and so it basically removes this arginine tag, and this albumin will basically become the mature form of the protein. And then for insulin, we see that before it becomes insulin, it's proinsulin, and it has to be, in order to become insulin, it has to be cut by two different proteins here called PC3 and PC2 endoprotease, which will look for this arginine and lysine-arginine sequence, and they cut the sequence at this point, and you can see that this part is left in this blue part as well, and a carboxyl peptidase could further process this to remove these arginines, and now the insulin could be bound by these sulfur interactions. And then another important aspect of the late secretory pathway is studies with epithelial cells, which show how proteins are transported through the basolateral and the apical areas of the membrane. And so here this epithelial cell is made to synthesize VSVG protein from a virus, and also another viral protein, influenza virus, glycoproteins. And the way that they are sorted in the Golgi, in the trans-Golgi network, is some of the basolateral proteins will, along with some apical proteins, they'll be transported to the basolateral membrane. And the purple one is a basolateral protein, but the yellow protein here is actually meant for the apical membrane. And so the way that the apical protein is returned to the apical membrane from the basolateral membrane is through endocytosis of these clathrin-coated vesicles. And sometimes if the apical membrane protein is transported along with a basolateral protein, the basolateral protein could be recycled through budding off of this vesicle back to the basolateral membrane, and the apical protein will make its way back to the apical plasma membrane. And sometimes apical destined proteins directly go to the apical side. Yeah, and so this is showing that sometimes if proteins from the apical side are missorted to the basolateral side, they could undergo endocytosis and transport back to the apical side through transcytosis.